Hi, so I recorded this video in order to demonstrate what is the dynamics that one needs to follow when we are trying to solve questions such as identifying the distance in terms of genetic distance of map units between two or more different genes, right? Each with their different parental types. And for that, I'm taking material from the tutorial number 7. And it says that an Arabidopsis geneticist crosses two mutants PE, that stands for many petals, to a mutant for the gene SC, which stands for small cells. And the F1 heterozygote looks wild type. So what this means is that basically the reason why they cross the PE with the SC from the parental breed with each other to create an F1 was to determine the dominance of the different alleles that are in play. So basically, this is gonna be how the genotype of our parentals, those for the small cells, and those for the many petals is going to look like. So at this point, we don't know which one is which, right? But because they are pure breeds, we know that they must be, because they're pure breeds, they are homozygous, right? And because they only express one of two traits. For example, they are either they either have many petals or they either have small cells. We know that they must be homozygote for one of their respective mutations, whereas on the other one we have the wild type, right? So at the moment when we make this cross, we need to what we identify is that the output is a normal looking plant, right? Which means that there is complementarity. What this means is that the wild type for the many petals and the wild type version of the small cells is fully functional and takes over the phenotype of the individual. From this, we can say that PE plus is dominant over PE minus, and that SC plus is dominant over SC minus, right? So now we can start labeling which parental is which in this scenario. So this one has small cells, and we have individuals that are homozygote, recessive, and for one of the two genes of interest, the one for small cells. Therefore, this is gonna be the SC mutant, right? The other one is gonna be the many petals mutant. So this is how our parental breeds genotypes looks like. So the first question that we need to ask is how many different gametes will each of these parentals produce, right? And the correct answer is, well, we just need to play with the different combinations, right? At the moment of recombination, we have one chromosome with two different genes, one for PE, one for SC, and even if recombination happen, we're gonna have four different gametes. They are gonna have, but they are all gonna be PE plus SC minus, right? So we take all the different versions of PE for this parent, and the only one that we have here is PE plus, and then this PE plus can either go with this SC minus or this SC minus, but in terms of our nomenclature, they are identical. Therefore, that means that the only gamete that this parent can produce is of one type, and the same logic applies for the other one. So these are going to be the gametes that each of the parents can produce: PE plus SC minus and PE minus SC plus, right? So we have our the hybrid individual, and it is the hybrid because it's heterozygote for each of the genes of interest, for the PE gene and for the SC gene. But this individual, just as a reminder, is fully wild type, right? It has normal amount of petals and normal size cells. So that's why I mentioned that complementarity happened there. So an interesting question is how many different gametes will this individual, the hybrid individual, produce, right? So now we play again with the probabilities. Well, one gamete can be PE plus, the other one PE minus, right? And then this is one gene, and then we check the probability that this one goes with the respective SC plus or SC minus, right? Each of these, all of these scenarios are possible. Whether or not they are equally probable will depend as to whether or not these genes are linked or not. So this would give us the different, the total number of different gametes that this individual can produce, and in this case, well, it would be 4 different gametes, right? So that is going to be an important point to keep along while we start talking about how we calculate recombinance versus the parental types. Okay, so here we have the information that our dihybrid individual has been crossed in the F1, has been crossed with one of the double recessive parents, which means that the only gamete that this parent can produce is going to look like this: P minus, SC minus. Okay, and from this one we have the 4 different gametes: P plus, SC plus, PE plus, SC minus, and the complement, right? P minus, SC plus, and PE minus, SC minus. Okay, so those are the 4 different gametes. So each of these can go in combination with the only gamete that this other parent can produce, right? So this is what we call a test cross. And the reason why we call it a test cross is because the only genetic element relative to the contribution of the 2 parents, the F1 and the double homozygote parental, is going to be the only thing that is going to define the phenotype is going to be the contribution of the dihybrid parent, right? Because this one can be considered as a blank, some sort of like some sort of like a template against which the gametes from the dihybrid can be expressed. Let's look at an example. So I'm going to erase this for a minute. Okay, so they're giving us a table here. What is the genotype of each of these different offsprings? That is really the most important question. And what genotypes come from the parental chromosomes found in the F1 dihybrid? So, so here we have the table that summarizes the different offspring that we obtain. And in that different set of phenotypes, in this table, we have contained information on what was the contribution of the hybrid parent. For example, if we have wild-type individuals, the contribution of the hybrid parent must have been PE plus and SC plus. And the reason why I know this is because the only contribution of the double homozygote parent could have been PE minus, SC minus, right? At the moment that these two get combined, the positive, but the wild-type versions of the respective genes save the day, kind of thing, if we can say it like that, and return the phenotype of the individual to a wild-type for each of the 2 different genes. How about for many petals? Well, for many petals to occur, we know that this mutation is recessive relative to the dominant allele, which is the wild type, and the baby must have this phenotype, right? A double homozygote recessive. Therefore, the only gamete that defines whether or not the gamete is in homozygosity is the contribution again of the hybrid parent. So we have PE minus PE minus. Okay, but we also perceive that the number of cells, the size of the cells, is normal, which means that the hybrid parent, the F1, must have also given an SC plus, right? Otherwise, because the double homozygote parent can only give this gamete again, these plants would have also had smaller cells. Okay, so we keep applying this logic down the road so that we can end up getting this table right here that summarizes the genotypes of the different plants, right? And if you notice, there are constant values here. In all cases, these plants are going to have Pe minus, at least one of their genes being Pe minus, and at least one of their SC genes being having the allele minus as well, right? So these are the contributions of the double homozygote parent. So you could even just, get rid of it so that you just get the information on the F1 contribution. And the reason why I'm doing this is because, well, from the genotypes of the gametes that the F1 individual produced and provided to their offspring, we get information of which ones are recombinants and which ones are parentals. So we know that one of the parents must have been Pe minus SC plus, and the other one P plus SC minus, right? In terms of the gametes that each of the original parentals gave to the F1 individual. So in the cell, in the somatic cells of our F1 individual, you're going to have 2 copies of each chromosome, and then you're going to have here one being P minus in combination in this, what I mean is in the same chromosome to the SC plus. Right? And the other one, this one came from one parent. The other chromosome that you can find in that cell is going to be PE plus and PSC minus. Okay, so these are the parental configurations, the parental gametes, right? If they're passed to a meiocyte during the stage of meiosis as such, it means that those were the parental gametes. If there is recombination happening between these two, then suddenly you create new combinations of gametes. In this case, P minus, SC minus, and P plus, SC plus. Okay, so these are recombinants. And which of these individuals in our table represent recombinants and which ones represent parentals? Well, now that we subtracted the information from our table, it's much easier to see. So the wild type is going to represent a recombinant. In the same way, individuals with the 2 mutations expressed are going to be also recombinant. So here we're getting evidence on the distance in terms of recombination frequency that these 2 genes are apart from each other, right? Based on the evidence of the amount of recombination that happened in the F1 parent of this test cross. This is a form of test cross, right? In order to identify the different patterns. So here we're going to have the parentals. These are going to be parentals, right? And you can go back and compare it. They're right here. So now we just need to count them. And if the ratio of parentals to recombinants is 1:1, in other words, there is a 50% frequency of recombinants, that means that the 2 genes are located in different chromosomes. And you can see it right here on the right. For example, if these 2 genes, SC and PE, start getting farther and farther apart from each other, there will be a point in which recombination will depend, the frequency of recombination will depend exclusively on chance. What is the probability that recombination will happen or won't happen? That will cut these 2 genes and take them to the other to the other homolog chromosome. What is going to be the probability? Well, it's just going to be 1/2. It's going to be 50%. It happens 50% of the time, right? Which is the same that happens when you have chromosomes that are with their respective genes that are completely physically separated from each other, right? The probability that this one is going to go with this one is the same as this one going with this one in the cell, right? So it's a 50%, is 1/2 for each of them. So it's the same thing, right? So you wanted to consider that as a recombination frequency, you would end up getting 50%. Now, that is different to say that the frequency of each recombinant is 50%. That is not true. The recombinant frequency of the parentals is often higher, right? And than the recombinants. And in the recombinants, the frequency in map units is going to be divided by 2. So if you have 50 map units, to each recombinant, you're going to assign a probability of 25%. And the same goes for the parentals, right? Each parental is going to have a probability of 25%. Okay, so during your tutorials and during your midterms and finals, you're going to encounter tables that are a little more difficult to interpret it. So the key and my suggestion for you is to look at them in pairs. Don't try to look at the data as a whole, but make comparisons. For example, now they're telling us that instead of crossing the petals with the small cells mutant, we cross the petals with a mutant for short roots. Okay, before we proceed to make the comparison to the new pair of mutants, we need to fill up the information that we already have. This is the information between the petals and the small cells. And there it is, right? So we determined that the distance in map units between the two different genes is of 50%. So now we can proceed and compare with the small roots genotype. Okay, so the many petals organism is going to have the same genotype as before. It's going to be PE minus, and then we're going to have a different gene. We are not playing with SC anymore, but SR. SR plus plus, right? Because these are purebreds. They're assumed to be homozygote for all of their genes. That is an important assumption. And then you're going to cross this with the individuals that are that have, so these ones have many petals, whereas these ones are going to have short roots. So that's how the parentals look like. And the reason why I know which one's which is, again, because they made an F1 cross, and again the wild type individual was the one that had the pattern that we observed before, right? The one that had only a double homozygosity was the one that was recessive. So if you keep playing with the data, again, you need to identify how many different gametes each of these parents produce. And again, it's going to be only one. PE minus is R plus, and PE plus is R minus. So those are the gametes that these parentals can produce. And they are going to give them as such to their baby, which is going to be called the F1. And we're going to have a chromosome that looks like this, PE plus, let's say SR, PE minus SR plus, this is for this parent, and SR minus PE plus. Okay, so that is the configuration of the F in the cell of the F1 individual. So now this baby plant is going to grow and is going to produce 4 different types of meiocytes, right? That's why you have 4 different elements here. So which genotype are going to correspond to which phenotype? So we are crossing again with double homozygote recessive parent, okay, for the gene of interest. So we are going to have, again, so these are going to be the parental gametes. So we have a PE minus, SR plus, and we have PE plus SR minus, which means that the recombinants are going to be PE minus SR minus and PE plus SR plus. Okay, so these are going to be the contributions of the F1 individual. So now we cross this with a parent double homozygote recessive that can only produce one type of gamete, PE minus SR minus. Okay, and then we do that to complete the total number of cells that are needed, so that in the new babies, so that we can observe the actual babies, not just the cells, but the babies that are diploid in and look at their phenotypes. So those phenotypes are going to depend on the dihybrid parent, right here. And then we can start looking at the relationship between the phenotypes and these phenotypes and the genotypes that the F1 produce. Okay, so for PE minus, we're going to have an individual with many petals and a wild type for the small roots. Okay, so that is this one, PE minus SR plus, which is parental. For PE minus SR minus, we're going to have an individual with many petals and small roots, which is this one. So that is going to be PE minus SR minus, which is recombinant. Then we have PE plus SR plus. That is going to be a full wild type individual. This is going to be PE plus SR plus, and that is a recombinant as well. Okay, and finally we have the other parental, which is PE plus SR minus, and that is going to be a parental as well. So now we have a higher frequency of the parentals and a lower frequency of recombinants. So it kind of sounds like these, there is something funny going on, right? Like there is some recombination happening. Some linkage happening. Okay, so what you need to do is just to calculate the total number of map units. Okay, and once you calculate the distance in map units, you should end up with something that gives you around 26 map units. Okay, so basically what you do is to add these ones together, the recombinants together, and divide it by the total. Okay, and that should give you around 26%. Now, what is the probability of observing each of the different babies? Well, it's going to be 26% divided by 2, which equals 13%. Okay, so I hope this helped you guys. I know I didn't really prepare a lot for this. This is old material, but I hope that it helps you. Now, finally, the last thing that you need to do is to calculate the frequency for the third combination, right? Which is small roots and small cells, and then you're gonna end up with the distance in map units for all the different combinations for your 3 different genes, right? Again, don't try to read everything at once. Try to take pairs and then calculate the frequency of each pair, and then you should arrive to something that looks like this, right? Where you calculate the distance in map units between the different genes and determine that from PE to SC there is no genetic linkage, even though there might be this might be physically linked, because PE to SC are physically linked and SR and SC are physically linked. Therefore, PE and SC should also be in the same chromosome, although they are so far apart that 50% of the time recombination splits them apart.